Hello everyone. Today my colleagues and I will share the results of our research project with you. Our study focused on the anatomy of two genera of orchids found in Thailand, Apostasia and Appendicula. Apostasia is a genus of 8 primitive orchids in the family Orchidaceae. These terrestrial, evergreen, glass-like plants are very recognizable as orchids and are distributed in humid areas. 3 species of Apostasia are found in Thailand. On the other hand, Appendicula, this genus is epiphytic, lithophytic, or rarely terrestrial herbs. There are 50 species of Appendicula in total, and 7 of them are found in Thailand. We conducted an online Google Scholar search, but lack of the research articles on the anatomy of this genera, which are relatively rare in the wild and lately extinct. However, both of them can be found in Khao National Park. Therefore, we decided to study their anatomy to construct the keys for species identification in case morphology or other science cannot be used for identification purposes. For our methodology, we used the peeling and krilling methods to study the epidermis and prepare transverse sections of leaf braids by using the paraffin method. We then stained them with safranin and fast grain. Our results show that the significant characteristic of leaf surface and leaf braids in transverse section for species identification were comprised of the shape of epidermal cells and cell walls. These features showed in 2 forms. For instance, Apostasia odorata has irregularly shaped cell walls, while Appendicula cornuta has polygonal shapes with straight walls. We also identified 2 types of stomata that can be used for species identification. For instance, Apostasia wallichii has parasitic stomata, while Appendicula hexandra has tetracytic stomata. Furthermore, we found that the shapes of the midribs can be divided into 2 forms. Apostasia wallichii has an adaxial surface that is convex and an abaxial surface that is concave, while Appendicula cornuta has an adaxial surface that is U-shaped and an abaxial surface that is concave. Finally, we identified the shapes of mesophyll as another important feature that can be used for species identification. In Apostasia odorata, the mesophyll has developed to become palisade and spongy, while in Appendicula hexandra, the mesophyll did not develop to become a palisade. In our discussion and conclusions, we found that the general anatomy of the genus Apostasia includes silica bodies and parasitic stomata in epidermis, which correspond to the descriptions of the leaf anatomy of the subfamily Apostasiae in 1992. On the other hand, the genus Appendicula features silica bodies in the stigmata, which correspond to the description of stigmata in Orchidales in 2008. The results obtained from this study can be used for species identification and represent the new report on the species studied. Thank you for your attention.